So in part 2 of chapter 8, we're going to look at how this genetic information is transferred from one cell to another. Okay, and we're going to do that in two different ways. We'll also look at the role of recombination between pieces of DNA and how that changes the characteristics of some of these microbes. This recombination can be involved. So we have two types of gene transfer. First, there's vertical gene transfer, and this occurs between mother and daughter cells. And so this occurs between cells that aren't around at the same time. So the mother cell, the bacterial cell, is going to undergo binary fission. And the first step in binary fission is the replication of the genome. So over here we see that there are two copies of the chromosome, here and here, and so DNA replication occurs to copy the genome, then everything else is replicated so the cell doubles in size, and then it splits in half, so cytokinesis. And then lastly, you have two identical cells. Now, if there was a plasmid inside here, it would also be replicated in that DNA replication, and it would be passed on to the daughter cell. So the mother cell is right here, the daughter cell is right there. And this is one of the major ways in which antibiotic resistance spreads, because if there is a plasmid in that mother cell, then you'll have two identical daughter cells that both are antibiotic resistant. So that's vertical gene transfer. In horizontal gene transfer, this is going to occur between cells that are around at the same time, and they can be cells of the same species or different species. Sometimes we get some transfer between different species. Okay, well, we've already talked about one of these ways that information can be transferred horizontally, and that's through conjugation. The other way, there's going to be a couple other ways we're going to talk about it. One of those ways involves a virus, which is shown here. Now, a lot of times what's being transferred between cells is going to be a chunk of DNA. Sometimes it's a chunk of the chromosome, but a lot of times it's going to be a plasmid. Okay, and there are different types of plasmids. The conjugative plasmids contain mating genes, so they allow these bugs to undergo conjugation. The assimilation plasmids are plasmids that usually contain a metabolic pathway, so they allow these bugs to carry out some sort of metabolism. A lot of times it's going to be some strange type of metabolism. So, for example, one of the bugs that you'll probably have a problem with when you have immunocompromised patients is Pseudomonas. Pseudomonas is a bug that has minimal nutrient requirements. It can pretty much make almost everything it needs in order to survive on its own. Okay, we've actually found Pseudomonas alive in distilled water, and really nothing's in distilled water but water. Okay, so that shows how minimal its nutrient requirements are. It can grow in a variety of different areas inside the body, and it can cause disease in those different areas. So it can cause wound infections if it gets in one of your cuts. It can cause pneumonia if it gets down in the lungs. It can cause septicemia if it gets in the bloodstream. Endocarditis if it gets in the heart. It can cause urinary tract infections. Just a variety of things that this bug can cause when it gets into those different places inside the body. There are a lot of different strains of Pseudomonas, and some of those strains contain a dissimilation plasmid that allow it to break down disinfectants called quats, and we'll talk about quats in chapter 7. An example of a quat would be Rogal, which is going to be one of the disinfectants you're going to use in the classroom. Okay, so this dissimilation plasmid allows the strain to not only break down the Rogal, but it also allows it to use that Rogal as a nutrient. And so when you're thinking, I want to spray this area with disinfectant to kill the bugs, and you are killing a lot of bugs. What you're really doing is you're killing off Pseudomonas' competition. A lot of times it's kept in low numbers because all the other bugs will outcompete it, even though it can grow in a variety of different places. Okay, but you're killing off its competition, so that means it can grow more. It doesn't have much to compete with. And you're also feeding it. You're feeding it by spraying along that row cow, okay, providing it with a nutrient. And lastly, we have the R factor. R factors, the R stands for resistance. So there's going to be resistance to disinfectants, resistance to antibiotics and stuff like that. Okay? Alright, and what really sucks is when you have combinations of these plasmids. So Pseudomonas is famous for having plasmids and having the ability to resist a variety of different treatments. Okay, you'll have a problem whenever you have patients like burn victims. A lot of them will succumb to infection because, especially if they're burned over a large area of the body, because they are really immunocompromised. Pseudomonas is going to be one of those main bugs that's going to kill them. So you don't want to spray that Rocal in that area. So along with having simulation plasmids, they'll also have a mixture of antibiotic resistance genes on those plasmids, and so they're going to be really tough to kill. And we'll also in chapter 20 talk about some other mechanisms of which Pseudomonas is going to be more resistant than some of the other bugs. We'll also be doing an experiment to test to see how resistant Pseudomonas is to certain treatments, and you'll see it is going to be more resistant than some of the other bugs out there. It's going to be really hard to kill. Alright, so those are the three types of plasmids. Those plasmids can be transferred a variety of different ways. One of the ways is through conjugation. And previously I explained conjugation. I actually explained how it occurs in a gram-negative cell. What they do is they use a structure called a pilus. And a pilus is like a harpoon. So when this cell that has a plasmid is ready to mate, it senses cells around it, shoots out this pilus, and the pilus contracts. And then when the pilus contracts, you can see in this area right here, there is a fusion between the cells. So there's an opening between them, and the cytoplasm is just confluent in each one. And so a copy of the plasmid will be given from one cell to the other. And then that cell lets go. So this will be broken and the two cells will separate. The same basic kind of thing happens in gram-positive cells, but instead of actually producing a pilus, what they do is they produce a sticky substance. So they get sticky on the outside, they actually just stick to the other cell, fusion occurs, and a copy of the plasmids being transferred from one cell to the other. Now these next couple of mechanisms involve a process called recombination. In recombination, what happens is you take two pieces of DNA and you just mix them up. You're going to break them, reattach them, and make different combinations. You can see up at the top here we have two chromosomes. Let's just say that chromosome B is the one that's already inside the cell. So you can see its genes are E, F, G, H, and I. And you can see it has capital letters for those genes. This bug just picked up a piece of DNA, which is going to be chromosome A. You can see it has the same genes, but they're alternative versions of those genes, which are alleles, different alleles. Okay? And so what will happen is inside the cell, there are a bunch of proteins and enzymes that have already been made that will line up these two pieces of DNA, and then they will cause a crossing over event to occur. They will break these pieces of DNA, and then they will reattach them. Now, a lot of times, one of these copies will be lost. So initially, we say that our chromosome B, as I said, had capital letters E, F, G, H, and I, and down at the bottom, we can see our recombinant has capital E, F, G, and then lowercase h and I. What a lot of people don't realize is that this is actually producing diversity in a population. So this bug was probably like all the other bugs before where it had capital E, F, G, H, and I, but now because it's recombination event, it is different. Its blueprint is different. Now it has a lowercase h and a lowercase I gene there. Now, is this good or bad? Don't really know. The bug actually doesn't even know. It's not like these enzymes that are causing this crossing over event to occur can read that DNA and say, okay, I want this gene, I want this gene, I don't want this one, let's just throw this away. Okay, they don't do that. It's just a random event. They line up that piece of DNA, they cause a crossing over event to occur, and then one of those copies is usually destroyed. So the bug is different to begin with. Now the advantage, there may be an advantage to being different. If you remember when we talked about evolution, we said there's a large population, genetic diversity, and selection. So when that selective force comes around, if it's a really strong selective force, maybe this bug is different enough from the other ones where it can survive. Because remember, the strong survive when the selective force comes, the weak are going to die. So these populations try and become genetically diverse so that hopefully the population survives when that selective force comes along. Over here on the left, we see another example. Let's just say that our original bug had this top blue chromosome, and this right here is a gene for penicillin resistance. So this particular bug is resistant to penicillin. Let's just say it's Staphylococcus aureus. Okay, very common for Staphylococcus aureus to be resistant to penicillin. Penicillin was started to be used actually in the 1940s on a massive scale during World War II. It actually saved a lot of soldiers' lives. We started seeing mass resistance in 1953 for penicillin. So it was about 10 years later, and we see massive resistance. And nowadays, over 90% of the strains of Staphylococcus aureus are resistant to penicillin. So that's not a good one to use if you have a staph infection. And so this bug only has one gene for antibiotic resistance, penicillin resistance. It picks up this yellow piece of DNA down here, and this yellow piece of DNA has ampicillin resistance, tetracycline resistance. And that kind of sucks. So when it picks up this extra piece of DNA, you line it up, there's a crossing over event, and there are two possible combinations. The first one, up at the top, you can see you have penicillin resistance together with ampicillin and tetracycline resistance. And so we're starting to make what we call superbug. It's resistant to multiple antibiotics. Okay, so that's one possible combination. The other combination you can see at the bottom has no antibiotic resistant genes there. So this bug is actually sensitive to all of the antibiotics, penicillin, ampicillin, and then tetracycline. Okay. Now in a population, what will happen if this chromosome is floating around? You can get both possibilities. It's not like I said, it's not like these bugs will read that DNA and say, okay, this is the gene for ampicillin resistant, tetracycline resistant, I want this, and I'm going to throw everything else away. They don't do that. Okay, it's just random recombination. You'll get both possibilities, and what will happen is when the selective force comes around, like penicillin or ampicillin or tetracycline, the bug on the bottom that carried out this recombination will die. The ones that are on the top that have this particular chromosome are going to live. All right, so our second way that we can transfer information horizontally is through a process called transformation. And transformation is the uptake of naked DNA. Okay, when I say naked DNA, what I mean is what's happened is a cell has lysis broken open and its DNA is just floating around in the solution. And so here's that DNA molecule right there just floating around and has certain alleles. And in transformation, these cells, certain cells become competent to take up that naked DNA. So you see this cell has uppercase alleles and this piece of DNA has lowercase alleles. So during certain times, they will become competent to take up that naked DNA. They take it up, recombination occurs, and in the end, we have a different strain. Because remember, our original strain had all capital alleles. Our new strain has a lowercase allele and three capital letter alleles. Okay. Now, as I said, there are certain times when these cells become competent to take up naked DNA. It doesn't happen all the time. And I'm going to show you when it does happen. So over here we have a growth curve. Okay, you can see the number of cells on the y-axis, time on the x-axis. And basically you can describe what happens when, let's just say whenever you inoculate a culture tube. A lot of times when we inoculate a culture tube, we start from a single cell. Okay, so we put a certain amount in there. And you can see how this line stays flat. Wow, cells really aren't increasing. They've entered what's called lag phase. Okay, they've been transferred from one environment to another, so it's kind of a little bit of shock. And what they need to do, they need to figure out if their minimal growth conditions are met. So that's what they're trying to do first. They're deciding whether or not their minimal growth conditions are met. So they have to look at the environment, sample the environment. You're using receptors on the surface to figure out whether or not it's good to go. And so after they decide yes, and then the second thing they're going to do in lag phase, they're going to prepare to divide. There's a lot of stuff that has to go on there to replicate the DNA, replicate everything else in order to divide. So that takes a little bit of time. And if they decide not to divide, then none of the other steps that I'm going to talk about will actually occur, and they won't increase in numbers. But conditions in the beginning are going to be very good. There's lots of nutrients, very little waste. So to decide yes, you're going to divide, start preparing, and then eventually they will start dividing. When they start dividing, you can see how there's a really rapid rate of increase in the number of cells. They've entered in the log phase. There's exponential growth here. So they divide very, very quickly. Their numbers increase very quickly. They go from 1 to 2, 2 to 4, and so it's exponential, and that's why that kind of looks like half of a parabola. Okay, and so there's a lot of metabolism going on. Because there's a lot of metabolism, these bugs are usually very, very sensitive to our treatments. So we're going to see in chapter 7, chapter 8 that our treatments a lot of times target metabolism. But as they metabolize and as they grow in numbers, what they're doing is they're using up the nutrients, producing waste. And so eventually conditions change. You've got lots of waste, little nutrients. And so they're going to have to slow down their rate of growth, and eventually it will kind of plateau and they've entered into stationary phase. And there are a lot of cells that are there. Some of them are dividing, some of them are actually dying, and that's why you get this leveling off effect. Okay, in stationary phase, there's tons of cells there. The amount of life equals the amount of death. And as they continue to live, the conditions start to get worse, or continue to get worse, because they're using up the little remaining nutrients and they're producing more and more waste. Okay? And then lastly, the last phase is when they pretty much just run out of nutrients and have a lot of waste. They enter death phase. And you can see how death phase is kind of like a mirror image of log phase. There's exponential death. And what that means is a lot of them die very quickly. Okay, so you have this sudden decrease here, and then the rate of death slows down towards the end. It's important that you realize that the weak die first, the strong remain behind. Don't forget that, especially when dealing with antibiotics. We'll talk about how that leads to resistance a little bit later. But in the stationary phase here, this is an important phase because they do some stuff a little bit differently here. Here, remember, there's a lot of life, but there's also a lot of death, and so they're trying to survive. They realize the conditions are getting worse, and so when they are in stationary phase, they'll do some things that they don't normally do, and one of those things is transformation. So remember, there's a lot of life and there's a lot of death. Those cells that die will life, so they break open and they provide other cells with their DNA. And those other cells in stationary phase become competent to take up that naked DNA and then undergo transformation. Now let's look at some conditions inside of your body. Think about when you go through when you eat contaminated food. Let's think about chicken, for example. In chicken, most of those chickens have been fed antibiotics their entire life, so the microbes on them are antibiotic resistant. And remember, we talked about pathogens. Less than 10% of those microbes out there are pathogenic. Okay, so you expect not over 90% of those microbes on the chicken to be non-pathogenic. But since the chickens have been fed antibiotics their entire life, those microbes that sit on the chicken are all antibiotic resistant. So even though they're non-pathogens, they still have these genes for antibiotic resistance in them. So you barbecue and you contaminate your chicken and you eat this contaminated chicken. Okay? Those bugs, when they're in your intestines, they enter the lag phase. Okay, first they decide conditions good to grow. And they say yes because there's plenty of food there. Okay, and then they start preparing. And then they undergo log phase, the exponential growth, along with the rest of your normal flora. They are dividing like crazy when there are tons of nutrients. Okay, but after a while, what's happening is they're using up the nutrients. You're actually absorbing nutrients as well because that's the purpose of eating. And so conditions start to get bad and they enter into stationary phase. Most of the time, in your intestines, you won't enter, the microbes won't enter death phase. And the reason why is because you've taken a dump, you've gone to the restroom, um, or you've eaten again. Okay, and when those two things happen, when you're removing waste, you're putting more nutrients in there, they go back to log phase, or lag phase, sorry, because conditions have changed. They will actually decide conditions are good and decide yes real quick, prepare, divide, and then they'll enter log phase. So it's constantly going from lag to logs to stationary. You eat again, you change the conditions, they go lag to log stationary. Every time they hit stationary phase, these bugs will some of them will live, some of them will die. Those that die release their DNA. And so with those chicken microbes that are antibiotic resistant, their genes for antibiotic resistance, when they die, their genes are going to be released. And then other things like E. coli will pick up those genes and assimilate them into their genome. And that's one of the ways that we get antibiotic resistant strains of E. coli. And what's interesting is the studies have shown that when new antibiotics are introduced into livestock, it takes about 6 months for us to start seeing resistant strains of E. coli causing urinary tract infections. So they monitor for the presence of those antibiotics in those urinary tract infections. And so I think they're looking specifically for that antibiotic resistance in things like E. coli because E. coli is the number one cause of UTIs. And so when they get data like this, what will happen is the FDA forces these manufacturers to remove that particular antibiotic from the actual feed, and it takes usually about what we've seen is about 2 to 3 years for the resistance to go down, resistant population of E. coli to actually go down, and we can start using that antibiotic to treat human patients again after a couple of years. Okay, but it takes a lot longer for the microbes to lose that resistance gene versus actually gaining it to begin with. Just that it takes about 6 months when they first start introducing the antibiotic in the livestock feed for us to start seeing those UTI that are caused by resistant E. coli. But a couple of years to get rid of that resistance gene. All right, our next mechanism by which genes can be transferred from one cell to another involves a virus. It actually involves a virus's mistake. So this is what a typical naked virus looks like. I know I've got some great artwork here. This blue little hexagon here is going to be a protective protein coat called the capsid, and it has a couple of different roles. First, it protects the genome. This red thing in here is a new NPC. It'll be either DNA or RNA. Okay, viruses don't like to follow the rules, so this is just nucleic acid. Okay, so it protects the genome of this non-living pathogen. Second thing it does, it attaches to a specific host. There are usually going to be little proteins on the surface of this capsid that will bind to cell surface receptors. And since specific cells have specific receptors on their surface, they have different receptors on the surface, only certain virions or virus particles are going to be able to affect certain host cells. They're usually very, very specific. And then once it attaches to the host, then it injects the genome inside the cell. Well, the viral genome can do a couple of things. It contains the information to take over the host cell, and it also contains the information to make more virions, or virus particles. And these things are really, really good at replicating. So here's my big host cell. First step is attachment. That capsid is involved in recognizing the host cell. It binds to receptors there. And once it binds to the receptors, it injects the viral genome. Now the virus takes over the cell. It's going to take over the central dogma. And when it takes over the central dogma, the DNA replication, it produces more genomes, which are down here. It produces more capsids through transcription and translation. It's going to be those protein capsids. The genome is then inserted into the capsid, and then the virions exit. Now, it's not just going to be one virus that's going to exit. There are actually usually hundreds to thousands of them that exit. They're very, very good at replicating. Super good at replicating. And so one goes in, hundreds, thousands, sometimes even millions come out of a single host cell. So basically, this virus is using that host as a factory, and the host isn't able to do what it normally does. And remember, this is very, very specific. There's going to be a particular type of cell that's going to be infected by this virus. So a good example of this would be HIV. Okay, HIV only infects a couple of types of cells inside of the body, mainly, and those would be the helper T cells and the macrophages. So it's very specific to those types of cells. Another example would be the cold and flu virus. Both of those like the cells in the upper respiratory tract. Okay? So all they have to do to find their host is actually get right inside your nose, and then they can start replicating in those cells that are there. And that's why the cold and the flu are actually really contagious. Not only that, but their infectious dose is really low. It's estimated to be less than 100. For the cold, it's estimated to be just a single virus. Okay, so our next mechanism by which genes can be transferred is transduction. And this actually involves a mistake that's made by the virus. So here's our virus, and you can see they call it a phage. Its actual full name is bacteriophage, and the bacteriophage infect bacteria. There's a specific type of virus that infects bacteria. So this right here contains viral genome and the capsid. The capsid binds to the host, injects the genome, and at this point right here, there are two blueprints. There's the bacterial chromosome here and the viral genome right there. In order to take over, what will happen is the viral genome will produce some enzymes that cut up this bacterial chromosome. So you can see how it's cut up into little pieces. You can see DNA replication has already occurred, so the viral genome, there are multiple copies of it. Transcription and translation will make the capsids, and then those genomes will be stuck into the capsids. Now you can see that most of these virions here are actually they actually look like the original virion. Now they have the viral genome inside of them, but there are some that are mistakes. Okay, there's one right here, and there's one right here. Okay. Instead of having the viral blueprint packaged into the capsid, one of these little chunks over here of chromosome, bacterial DNA, was stuck in there. And so it's a mistake. Now, remember the roles of the capsid? It protects the genome, it binds and recognizes the host, and then it injects DNA. And so these normal virus particles will go find another cell, and they will go through this whole process again, take over the cell and kill the cell. Okay, but these mutant ones, these defective ones that have bacterial DNA, all that can happen is the capsid can do its job, which means that it will recognize the host. Okay, it will attach to it and it will inject the genome. But this bacterial chromosome doesn't contain the information to take over the cell. It doesn't contain the information to make more virions. So all that happens is this DNA gets injected into the cell. Recombination occurs, and you can see how it's actually inserted into that bacterial chromosome. So now this new cell, a different strain, has some extra information, can do some extra things, but it depends upon what was in that piece of DNA, what genes were in that piece of DNA. Okay, so transduction involves a virus, and it's a mistake that's made by the virus. It just, instead of packaging its own genome into capsids, it packages a piece of the bacterial chromosome into there. And so transduction can occur anytime a virus is around, and it's going to be species specific. So remember, these virions, they will attack a certain type of host cell. And so if this is E. coli here, chances are this one's going to be E. coli as well. Sometimes they can attack a couple of different species, but usually it's really species specific. And lastly, this shows a nice summary of what we've talked about in Chapter 8. Over here on the right, we have what goes on inside. Information inside the cell stored in DNA. To make use of it, we have to go through gene expression, which is the sum of transcription and translation, to get those proteins which are going to do whatever they need to do for the cell. And one of the most important roles is catalysis, so we're talking about metabolism there. Okay? And so that's what goes on inside the cell. Transcription and translation are always going on as long as the cell's not dormant. In order to transfer that genetic information from one generation to another, we need DNA replication. Okay, so we go from one mother cell to DNA replication, replicate the genome, replicate everything else, and in binary fission, we get two daughter cells, each genetically identical. Now, if there was a plasmid in here, it would be replicated as well, and each of the daughter cells would get the plasmid. And so that's vertical gene transfer, going from one generation to the next. And remember what's happening, vertical versus horizontal. A lot of people get confused. Think of a family tree. So you go from one generation to the next. That's usually done vertically in a family tree. The other ways we talked about through transferring genes going to be through horizontal gene transfer, and that was conjugation, transformation, and transduction.